Hi guys, welcome back to my channel. Today we're going to do a passage from AAMC full length 2 biochem section. So if you had already done the test, this is for you. If you haven't, this could be a spoiler, or if you're welcome to just take a screenshot and give it a try before we dive into this. All right, we're going to do all 5 questions after we read the passage together. All right, so let's dive into this. So the passage reads, Ebola virus, Ebola, causes a rapidly progressing and often fatal hemorrhagic disease that currently has no effective treatment. Okay, so I know this passage is about some virus, okay? Ebola particles consist of a nucleocapsid encased in a membrane that contains transmembrane viral glycoprotein, EGP. The virus has a single-stranded negative sense RNA genome that is used as a template for the mRNAs produced by a virally encoded RNA-dependent RNA polymerase. The entry of an Ebola nucleocapsid into the host cell cytoplasm starts with the fusion of the viral membrane with the host cell membrane, a process facilitated by EGP. Recognizing the similarities between how EGP and the glycoproteins of related viruses are processed during viral infection, as well as how inhibitors of endosomal acidification block Ebola infection. Researchers hypothesized that acid-dependent proteases trigger the conformational change in EGP that are necessary for fusion. Okay, there's a lot of information from the first 2 paragraphs, right? As we actively read this, we want to have a mental pathway, what is going on, okay? So let's draw it out. So I have some virus, it's the Ebola, and then Ebola has some very important glycoprotein called EGP, right? Okay, so that we know that this virus want to infect my host cell. Okay, so this is my host cell. So how does it do it? How does it enter the host cell to infect the host cell is through membrane fusion, right? And then this fusion is facilitated by EGP, okay? And then we further know that the researchers hypothesized that acid-dependent proteases triggered the conformational change in EPG that are necessary for fusion. So now we know before EPG help with the fusion of the membranes, we have this proteases that will cause the conformational change of EPG. Then EPG can further help with the fusion of the membranes, right? So the virus can get inside a cell, okay? And then once it's inside a cell, the RNA-dependent RNA polymerase can transcribe the mRNA given the genetic info of that virus, which is RNA, right? And then later these mRNAs can be translated into viral proteins, then infection really happens, right? Okay, so just a little just a little connection to the first paragraph, the last part of first paragraph. Okay, all right. But we also know that from the second paragraph, there's a way to block infection, right? Here, inhibitors of endosomal acidification block Ebola infection. So that we know that this fusion can really be blocked, can be inhibited by what? By inhibition of this EA, okay? Endosomal acidification. Okay, this inhibition of endosomal acidification can really stop the fusion and then the infection, okay? So, all right, that's what we get from the first 2 paragraphs, okay? I mean, if you keep practicing drawing out the pathways when you are practicing, then hopefully on your real deal, on your MCAT exam, this becomes a second nature and you can just have this pathway in your head as you actively read the passage, okay? All right, let's keep reading. The infectivity of VSV, EPG, vesicular stomatitis virus, SVS particles engineered to contain EPG instead of a VSV glycoprotein in the viral envelope was reduced to more than, okay, what was reduced? The infectivity was reduced, okay? When we have this, when we get rid of VSV, when we have this EGP, the infectivity was reduced to more than 99-fold by what? By inhibition of the mammalian proteases CAT-B and CAT-L, okay? So now let's go back to our pathway and expand our pathway, okay? So now from the third paragraph, we know CAT-B and CAT-L, there are 2 proteases, okay? So that if we inhibit the inhibitors of mammalian proteases CatB and CatL. If we inhibit these CatB and CatL, these proteases, then this pathway of infection will be blocked, will be inhibited, right? So that's why it says, okay, the infectivity was reduced more than 99-fold by inhibiting those proteases, okay? All right, so that all makes sense, okay? All right, perfect. Okay, let's keep reading. In a separate experiment using a strain of Ebola, CatB inhibitors reduced the number of Ebola particle produced in cell culture tenfold, okay? So the number of Ebola particles produced in cell culture just is another way of saying infection. So this yellow sentence that I highlighted is just saying CatB inhibitors reduced the infection by tenfold. Okay, that also makes sense, right? You inhibit this CatB so that the protease would not be able to change the conformational change in EGP, and then the EGP would not be able to help with the fusion of the viral membrane and the host cell membrane. Then the virus will not be able to get inside my host cell, right? So no infection happens. The infection will be reduced. Okay, all right, perfect. So let's keep reading. Figure 1 shows how the introduction of certain genes into 2 mutant mouse cell lines affected the subsequent infection of these cells by VSV EGP. Okay, so this is figure 1. Okay, so the legend says figure 1, effect of CatB or CatL or both on VSV EGP infectivity. Okay, so we know the infectivity, the y-axis is the dependent variable of my experiment, right? Something I want to measure. I want to collect data from my experiment, right? So if the bar is super high, the column is high, then the infection is high, right? If it's low, then infection is low, okay? That's just the dependent variable, infectivity. And then also the independent variable of my experiment is really the effect of either CatB alone or effect of CatL alone or effect of having both CatB and CatL. Having both CatB and CatL, see how that will affect the infection versus having just CatB or CatL, okay? If that makes sense. Okay, that's the independent variables of my experiment, okay? All right, so usually we don't want to dig too deep into the figure unless we come across a question, right? But I want to really do a really general analysis of this figure, okay? That's what I do for all the figures I see on MCAT, right? I don't want to dig too deep, but I really wanted to know what are the outliers, those things that strike out, those super tall bars, what do they mean, right? Before I jump into the question, okay? So let's see. So I know that from my figure, I have 2 mutant mouse cell lines, right? So I do see this is a wild type. I didn't do anything to the gene, right? But here I have 2 mutations. This is the first mutant cell line. This is a second mutant cell line, okay? So the mutation of the first mutant cell line is I got rid of CatB. CatB gene is knocked out, right? Minus, 2 alleles, right? So both alleles got knocked out, meaning that I'm not going to have this protease CatB getting translated into protein, okay? So that I don't have a CatB. I only have a CatL, okay? And then the second mutation is I knocked out both genes, okay? I won't have neither CatB protease nor CatL protease, none, okay? All right. Okay, so that we see that without knocking out any gene, I have a pretty high infectivity, right? I have a pretty high bar because this wild type is really, I have both CatB and CatL. I didn't knock out any gene, right? Okay, but I see that there's 2 special cases here. These 2 bars are pretty high as well, right? Okay, what is going on here? That I see that, okay, I don't have CatB. I do have CatL, but this culture cell without CatB, I introduced CatB. Okay, I don't know why they do that, but that's what they did, right? Okay, they knock out CatB, but then they introduce CatB. Meaning what? Meaning with this column, let's label 1, 2, 3, 4, 5, 6, 7, 8. This column 3 is just like column 1. It has both CatB, and CatL. Okay, I did knock out CatB from the get-go, but then I introduced CatB, so that's why I have both CatB and CatL. And that makes sense that its infectivity is high, right? Because I have both proteases that can facilitate infection. Okay, all right. And then what about the column 8, right? It's also it strikes out because it's also relatively high, even though I knocked out both proteases, right? Okay, then we know that the cell culture that we don't have both genes, but then we introduce cat B and cat L. Okay, so column 8 is just like column 1 and column 3 that we have both B and L. Okay, so my very general, very quick analysis of this figure is that I know to have the highest infection, to have the to have decent amount of infection, if you will. I shouldn't say decent, but anyways, to have great amount of infection, we have to have both cat B and cat L. When they coexist, when we have both of them, that's when we have the highest infection. Okay, so that's it. That's my level of understanding. Okay, as I was reading the passage, and I don't want to dig too much deep into every single column. If I do need to analyze it, then I'll come back. Okay, all right. Let's jump into the question. Okay, so the first question here says, based on the passage, cat B and cat L most likely act on EGP in which of the following cellular compartments to facilitate membrane fusion? Okay, so let's go back to our pathway that I highlighted in pink that I know, those cat B, cat L, they're proteases, they change the conformation structure of my EGP, and then EGP can facilitate the fusion of the membranes, and then voila, the virus is inside the cell, then infection can happen, right? But we know that there's something that's blocking, this infection. Okay, what is this? It's the inhibitors of endosomal acidification. Okay, inhibitors of endosomal acidification block this infection. Okay, you might need a little bit background knowledge. Okay, about endosome. So endosome is an organelle. Okay, it's inside your cell, and then once through the endocytosis, once the fusion happens, then you have this virus, this Ebola inside your host cell, right? And then they will get into this structure organelle called the endosome. Okay, and then the endosome is gonna later mature into lysosome, so that we know lysosome is really acidic dependent, right? It says low pH, very acidic, and then it has a lot of enzymes can degrade the virus. That's the ideal situation if we don't want to get infected. Okay, if there was no infection happening, then even if the virus gets inside the cell, the virus will stay in the endosome, and then later the endosome mature into lysosome where the virus will be degraded. Okay, so that is no infection. Okay, so the passage says inhibitors of lysosomal acidification. So what is a lysosomal acidification? EA. What does EA mean? Endosomal acidification. Endosomal acidification basically means no endosome, right? Endosome gets acidified, so there's no endosomal activity. Okay, so if we don't have endosomal activity, if endosomal acidification happens, then infection will happen because endosome cannot, it's acidified. There's no way it will mature into lysosome and degrade the virus, right? Okay, so inhibitors of EA means no EA. Okay, so no EA means no endosome degradation. So you do have endosome, okay? So no EA, or AKA inhibitors of endosomal acidification means what? Means it will be mature into lysosome, and then later the virus gets degraded, so no infection. So that's why the passage says, inhibitors of lysosomal acidification stop the Ebola infection. Okay, that's how it happens. It's all because the enzymes inside lysosome is gonna chew up all the viruses. Okay, all right. So that's how you prevent infection. Okay, so proteases and EPG, they want infection to happen, right? They're facilitating the infection. They're facilitating fusion and then infection. So which cellular compartment do you think cat B and cat L most likely will act on EPG so that this fusion, this infection can happen, right? Of course, it's the endosome, okay? Because with the endosome, if there's no endosomal acidification with endosome, then you can have infection, but we want fusion and infection to happen, right? Because that's the job of cat B and cat L and EGP. So we're going to choose C. Okay. All right. I hope that's clear. Now let's move on to the next question. Okay. Next one is overwhelming. Okay. Next one says, based on the passage, is cat-L expression sufficient for VSV EGP infection of the mouse cell lines presented in figure 1? Note, in these experiments, assume that values of smaller or equal to 3% of total cell infected are too low to be measured accurately. Okay? So basically, this worthy question, let's simplify it, is asking, is cat-L, this one of the proteases that was introduced in the passage, is this cat-L protease efficient for decent amount of infection, right? That's all it's asking about, okay? So I'm going to analyze all A, B, C, D for you in just a second, okay? I'm going to analyze it in detail, but just to show you how I do it quickly on MCAT, if it was my real deal, okay? I don't have time to read every single choices. And through my general analysis of the figure, I know that the tallest bars, the highest infections are occurring when we have both cat B and cat L. So I don't think just having cat L is going to be sufficient for infection to happen, okay? Because I just see other bars are being super low, okay? Meaning that the other bars could be the case of only have cat L expression, right? I don't think that's efficient for infection. Okay, so I'm going to cross out A and B because A and B says yes, right? But my assumption is I think I need both. Okay, so let's read C and D. So if it was my test, I'll read, okay, C says no because VSV EGP does not infect cat B minus cells expressing cat L better than it infects cat B minus cells not expressing cat L. I mean, I like it. Logically, it makes sense, right? Because it's saying that the cell that is expressing cat L is not doing a better job of infection, than the cells without cat L. So cat L is probably not sufficient, okay? So C is logical, okay? C is logically legit, okay? I mean, what about D? D, I would read no, because VSV, EGP infects cells expression both cat B and cat L better than it infects cells expressing cat B only, but not cat L, okay? So D is saying I need to have both B plus L to do a better job of, to infect more than just cat B does, okay? So D is telling me, the answer choice D is telling me both B and L together, that's efficient for decent amount of infection, okay? So it doesn't answer my question. D is not saying that, okay, D is saying B and L are sufficient. Both are sufficient. It's not saying that cat L expression is sufficient, okay? So, I mean, the question is really asking about cat L. So I still think C is my better answer, okay? Okay, that's how I do it quickly, right? On MCAT, I didn't bother reading A and B, and then D is just logically doesn't align with what the question is asking for. So I just chose C, right? But we can really dig deep into this, okay? So let's see. So let's analyze C first, okay? C says, because this VSV EGP does not infect B minus expressing cat L, okay? Better than it's comparing those B minus expressing cat L versus the cat B minus not expressing cat L, okay? So let's color label everything, okay? So from my figure, I see that cat B minus, okay, all of them except the wild type, right? Column 2 to 8, they're all cat B minus, okay? They're all cat B minus, but which ones are expressing cat L? Okay, I think this one is, right? Because I have a cat L plus here, okay? So this bacteria, okay, without this bacteria being the host cell, right? I didn't introduce any extra genes so that I don't have cat B and I do have cat L. So this is the first green one that I highlight, okay? Let's see if I have some other one, okay? So I think this one is also the one that do not have a cat B LL, that do not have cat B, but it does have cat LL, right? Because I didn't knock out cat L from the get-go, and then I also introduced more cat L. So this one have cat L, but it doesn't have cat B, right? What about column 3? Column 3 is not cat B minus, even though in the beginning I knocked out cat B, But later I introduced cat B. So really column 3 is the scenario where I have both B and L. Okay, so I'm not going to highlight column 3. Okay, let's see if I have another one that fits the category of cat B minus minus expressing cat L. Okay, I think this one, right? This one. Okay, I knocked out both, but then later I introduced cat L. Okay, so that I do have cat L. Okay, but I don't have cat B. Okay, all right. Let's see. Let's highlight the red one. Okay, cat B minus minus that do not express cat L. Okay, so I see only this one. Okay, this only one is it lack both genes. Okay, whereas the other genes, the other columns all have either cat B or cat L. Okay, or both. Okay, all right. So to compare all the green ones with the red one, do you see that the green ones are doing a better job infecting than the red one? No, right? The green ones are not taller. The bars are not taller than the red one. So that C is correct, right? Because that I do see that the green ones, the cat B minus minus cells expressing cat L is not doing a better job at infecting, right? Than the cat B minus minus cell not expressing cat L. Okay? I hope that makes sense. Okay? So that's my red and green. Okay? So now let's analyze another one. Let's analyze A. Okay, so A says yes, because VSV EGP infects cells expressing cat L. Okay, so I assume this is expressing only cat L. Better than it infects cells not expressing cat L. Okay, so not expressing cat L. Okay, so now let's highlight those. Okay. So let's first highlight the cells that's only expressing cat L. Okay, so I see this one, column 2 is expressing only cat L, right? Because cat L was not knocked out. Okay, and then which other one? This one, right? This one I also just have cat L because I don't have cat B, right? Cat B is knocked out. Okay. All right, I don't see anything else that I should highlight in yellow. And then what about the blue one, right? The cells that are not expressing cat L. Okay, so I see this one. This is not expressing cat L, right? And then this one is also not expressing cat L. Okay, so now let's compare the yellow ones with the blue ones. Okay, so do you think the yellow ones are doing a better job than the blue ones, as choice A says? No, right? We see that column number 6, one of the blue ones is has higher infection, better infection than the yellow ones, right? So A is just wrong, right? A is just wrong in all ways, okay? All right. What about B? B says, yes, because VSV EGP infects cells expressing cat B better than it affects cell not expressing cat B. I don't like this from the beginning because it's telling me cat B is so great, right? But even if it's true, even if we were to highlight answer choice B, even if it was true, it's telling me, oh, cat B is so great. But the question is asking, is cat L sufficient? Right? So, I mean, it's really out of scope. Okay, so B is wrong. Okay, so after this detailed analysis, still C is our best answer. Okay, all right. Next one says, based on the passage, does optimal VSV EGP infection in vitro require cat B, cat L, or both? Optimal infection, highest infection, right? Highest bars, of course, it requires both cat B and cat L, right? After the last question, this should be easy. Okay, perfect. Okay. Now let's move on to the next question. We have 2 more to go. Okay, based on the passage, cat B or cat L or both would be expected to have which of the following effects, if any, on EGP? Okay, so this is just going back to our pathway, right? So A, I can cross it out, no effect. Of course, there's effect. If there was no effect, why does the passage bother telling me about this pathway, how this proteases can affect the conformational change of EGP, right? So I don't think A is correct. Okay, what about B? B, reduction of enzyme activity. I mean, I don't know if EGP is an enzyme or not. I know it's a glycoprotein, but that's all I know, right? So I don't know. I mean, maybe not, or let's not cross it out so quickly, okay? Maybe not. Let's see if there's a better answer, okay? C says formation of protein dimer. This just sounds out of scope, irrelevant to me, okay? I mean, how do cat B and cat L form protein dimer? I mean, does forming protein dimer, would it help EGP, right? I mean, I doubt it because proteases Cat B, Cat L, they're proteases. Really, their job is chew up the proteins, right? They cut them short, right? Through hydrolysis of the polypeptide chain, right? So I don't think it's going to do anything other than what D says, digestion into smaller protein fragments. That's what proteases do, right? And Cat B and Cat L, they are proteases, okay? So D is my best answer, okay? So I can cross out B now, okay? All right, because B is just ambiguous. I don't know if that's an enzyme or not. But I do know that proteases do digest the proteins into smaller protein fragments, okay? So D is my best answer. All right, the last one. The precursor of EGP is translated from a transcript. Transcript is just mRNA, okay? Yeah, that has had one non-templated nucleotide added to the open reading frame, okay? This change does not create or eliminate a stop codon. Compared with the protein SGP, which is produced with the unedited transcript, okay? So this is the good protein. This is the unedited. This is the good mRNA, right? Okay, so the bad one, the mutated one compared to the good one, it's asking us what's the difference, okay? What's the difference? All right, so this is obviously a frame shift mutation, right? So I can draw it out for you. So I can just come up with an example. For example, I have a originally before the mutation, I have a like AUG, or let's just come up something random, or... Okay, so originally my reading frame is 333, right? 3 nucleotides, 1 amino acid, right? Okay, so that's my reading frame. Okay, now I added 1 nucleotide, right? Which is, I added 1. After I add 1, it's gonna change my reading frame. Okay, how so? I'll show you. So if I add 1, let's say, and then let's say here, I add another extra 8 here, okay? So now my new reading frame is like this, and this, right? It was here, right? So that you see that originally I have this stop codon, but now I added 1. It doesn't like create a new stop codon, and it doesn't eliminate a stop codon, right? I can still translate this protein given this mRNA, right? So this is really a frame shift. You shift the frame, but it really does change the it really does change the C terminus of the sequence, but the N terminus really doesn't get changed, right? Because the protein is still transcribed, okay? All right, so that, translated the protein. So let's read the answer choices. Amino terminal is just N terminus, right? Amino terminus is the same as the good one, but a different primary carboxy terminal, this is just C terminus, right? Yeah, it is different, right? Because I mean, yeah, it is different now, okay? I didn't create a new stop codon, and I didn't eliminate a stop codon either. So this is this bizarre situation, okay? So A is my best choice, okay? My best answer choice, okay? So let's B is just opposite of A, right? What about C and D? Let's just for completion read it. Sequence as SGP, except that EGP has 1 additional amino acid. No, I don't think so. Like to have one additional amino acid, like I said, you need 3 nucleotides, right? And then if you want to have 1 less amino acid, you need 3 less nucleotides. Okay, so all right, so this is this passage. It's pretty overwhelming. It's not very straightforward, okay? And then also regarding the last question, I want you to remind yourself what are point mutation. That's another high yield mutation, okay? That's different than the frame shift mutation in this question, okay? Point mutation is you replace a nucleotide with another nucleotide. So you don't add or you don't insert or get rid of one that caused the frame shift, but it's some other mutation, right? Point mutation includes silent, nonsense, or missense. Okay, all right. So this is this passage. If you find it helpful, please like, share, and subscribe. Thank you.